All right, everybody, we're going to go ahead and get started. Thanks so much for being here today at our research seminar. For those folks that are online, if you need any additional support, you can put into the chat box. We've got a volunteer here that's watching those chats. Her name is Roseanne, and she'll get back to you if there's any issues there. For folks in the room, thanks so much for being here. It's a little bit cold, but I'm going to celebrate something with my regulars. They've turned the fan down. So they turned this fan down and the room is colder. Yeah, but we're getting there slowly but surely. All right, everybody. Again, thank you so much for being here. Remember, this is a hybrid event. So if you have any questions for our speaker or for myself, please make sure to raise your hand at the end of the presentation and I'll bring you a mic. I need the reason to run, so go ahead and do that. Or you can go up to the mic stand over there and ask your questions. That way folks online can hear. For folks online, we do have your mics, cameras, and screen shares turned off. So again, if you have any questions, use that chat box and Roseanne will read them out to our presenter today, and we'll get those questions answered. Couple other quick little logistics. Wanted to let you know that today's event is being recorded. So if you'd like to share it with others that weren't able to be here today, it should be posted in about a day or two on the Hatfield website and past seminar page. So you can go ahead and find it there. Wanted to make a quick announcement for next week's seminar. That will be November 13th. 10th. Anyway, next week, Will and I are nods together here. We'll figure it out. We have John Stepnick from the Hacker Lab on main campus. That's going to be here talking to us about carbon storage in non-native beach grasses. So that is going to be kind of an interesting topic that I'm excited to learn more about. If you want to learn more about any of HMSC's events, you can go to HMSC, Google it, Firefox it, whatever you use. Scroll to the bottom of the page on that homepage, and there's a calendar of events there with all the login information that you might need. But the reason we're all here today is for today's speaker, and I'm going to hand it off to Will White, who is going to introduce today's speaker.
Thanks, Simon. Great. Well, today I'm really delighted to introduce Dr. Jess Hopf. Jess is a research associate who's been working in my research group for the past 2.5 years. And she lives and works in Queensland, Australia. So we're really delighted to have her flown all the way from Australia just yesterday to give this seminar. But she'll also be here resident at Hatfield for the next about 2 weeks. So lots of opportunities to ask follow-up questions and say hi to her. I've known Jess since 2016 when I was one of the external examiners for her PhD dissertation, which was all about, which is at James Cook University, also in Queensland, and all about the fishery management of the coral trout on the Great Barrier Reef. The coral trout is not a trout. Australians like to give fish funny names. It's a grouper, but Australians also call groupers gropers, and so maybe groper. Yeah, so we'll just say coral trout. But it was a really fascinating dissertation that looked at the way spatial management and marine reserves could be used to affect the fishery of that really important fishery species in Australia. In the years since then, I kept trying to find ways to work with Jess and build a collaboration. And finally, at the beginning of 2020, we worked out an arrangement where she would be able to work remotely and be part of the research group that way, which seemed like a really unusual working relationship at the time. And then it became the pandemic, and then it was just everybody was just a little head on a TV screen, so it was fine. And so Jess has been really productive working in our group. She's had a number of papers come out just this year in journals like Ecology Letters and Ecological Applications. And then she worked with us in our group's assessment of the Oregon Marine Reserves Program just this year, and in fact was the lead author of the report that our group submitted with recommendations and legislative proposals to the Oregon legislature and to the Oregon governor's office just last month. So I'm delighted to have her here and speaking about some of the work she's been doing on marine reserves in a changing climate. I guess I don't have it.
Thank you. Thanks, Will. Great. So I'm Jess. Hi. So I just want to first of all acknowledge that, yes, I'm far away from home, but I do work on and live in the lands of the Gubbi Gubbi people, which are the traditional owners there, Australian Indigenous people. And I just want to make recognition of that, that even though I'm far away, that's where I reside and I recognize that. So like Will said, I'm going to be talking, kind of giving you a bit of a snapshot of the work that we've been doing over the last couple of years. There will be a picture of a coral trout as well, so you get to see what they look like. They're quite mean and grumpy. They have like an underbite and they always look really frumpy. So I'm going to talk a little bit about that work as well, but it really is just a snapshot. So if you have any questions or anything you want to talk further, please feel free to come and say hi either today or over the next couple of weeks. I also done this work with Will, obviously, but also we've been working a little bit with Jen Cassell, who's at UC Santa Barbara. So I just want to acknowledge that too. Cool. All right. So marine reserves, or sorry, marine protected areas, also known as MPAs, are everywhere around the world. About 8% of our oceans are currently protected within an MPA, and there's big calls to increase this to 30% by 2030, which really isn't that far away. About 3% is within what's called a no-take area, also known as a reserve. So a main difference between a no-take area and an MPA is that they are a type of MPA, but they're a type of MPA where you're not allowed to do any sort of extractive activities like fishing or harvesting, or any destructive activities like ocean development. So they really are, truly protected areas from human exploitation. And their goal essentially really is to protect and conserve biodiversity. And this goal is often achieved through rebuilding populations or habitats that have been previously exploited in that area. And what this exactly means can vary place to place, and the specifics are sort of dictated by the local governing body and sometimes includes community consultations as well. So I hope you're aware you have reserves here in Oregon. There's 5 of them off the coast. And as Will just mentioned, we were just part of an assessment team of looking at these 5 reserves. They were established in 2012 to 2016. And prior to that, there was a reserve network that was established down in California as well. And both of these reserves have gone or are just undergoing assessments. And what we're really sort of looking at when we do an assessment is, what do we see going on in the reserves? Are they meeting the goals and expectations that we have for them? And then how can we, with this information, how can we adaptively manage these areas as we move forward in the future? And I was going to do a plug here for the State of the Coast, which is a conference on here on Saturday, but I just found out it's full. So if you can't come to that, sorry. But if you are going, we are talking about that review process on Saturday afternoon. So these reserves are under review. Also, apologies if you can't understand a word I say because of my accent, but hopefully it's getting through. But they're also, like many of the areas around the world, are facing increasing climate pressures. So I really like this diagram from the IPCC that kind of just gives a nice overview of all the climate related effects that are happening in the ocean, anything from heat waves through to ocean acidification. The aspect of climate change that I'm really interested in is the increased extreme events, or I more commonly refer to them as disturbances through this talk. So this map shows all the extreme events that have happened around the world since 1998, and there might have been multiple of these events in a given area. And the one that you're probably most familiar with is the marine heat wave, the blob, which I thought was a fantastic name for it, that came across in 2013 to 2015. And during that time, we really saw some dramatic changes along the coast. So for example, in Northern California, we saw nice, well-established kelp forests go to switch to urchin barrens within the space of a couple of years. And this has huge impacts. These sort of extreme heat events have huge impacts, not only for the kelp, but obviously for those species that rely on these kelp habitats for food. And the reason that I think about these events a lot, and the reason that they're really important to me, is because they're sudden and abrupt. They bring about sudden and abrupt changes. And these sudden abrupt changes not only impact the ecosystem that they affect, but also the communities, the coastal communities that rely on those ecosystems for livelihoods, for food, or might have some sort of spiritual or cultural connection with these ecosystems. So for example, when we saw the blob come through in combination with other local scale events or stresses, it led to the closure of the recreational red abalone fishery in California, and that's closed still for another couple of years. So these really bring about dramatic changes and have a real sense of urgency about them. And we need to think about, what are we going to do moving forward when more and more of these events happen? So bringing it back to reserves, which is what I do all my work on, what happens when we have an extreme event in an area that has a reserve, and why do we really care about it? So I wanted to talk, or disturbance, I'll also refer to extreme events as. So what I wanted to start off talking about was what we expect to see when we protect an area, when we put it within a reserve. So within the reserve, this is from California, within the reserve, we expect to see biomass or density of targeted fish species go up over time, which makes sense. You protect something from exploitation, it will rebuild. And then it'll rebuild compared to some sort of area outside or before the reserve was established. And this is one of our goals of marine reserves. If we're going to protect biodiversity, we need to rebuild things that have been previously exploited. When we have a disturbance, this might sound really obvious, but I wanted to point it out. It affects the species and populations both within and outside the reserve. So for example, during the heat wave, we saw cool affinity species such as your rockfishes. They declined both within the reserve, which is the green ones, and outside the reserve as well. And so the question is, okay, these areas are affected by these disturbances, obviously, but what does this really mean? And these disturbances kind of present challenges for the adaptive management of the reserves, and there's two sides to this. They first of all challenge what our goals and expectations are for reserves. So like I mentioned, our goals are usually to protect and conserve biodiversity, but what does that actually mean in the face of climate change? The other thing that they challenge is our ability to make accurate assessments of marine reserves. So if we expect one thing to happen in a reserve and then a disturbance comes along and affects that, how do we know that what we're looking at is the result of the disturbance or the reserve? So these two things go hand in hand, they're linked. We need clear goals and expectations in order to make accurate assessments. And then we also need accurate assessments to update our knowledge and our understanding of the system so we can feed that back into our goals and expectations moving forward. And this is really part of the adaptive management process. We need that toing and froing and that linking between goals and accurate assessments to help us achieve what we want to achieve out of reserves. So the two key questions I'm going to be talking about today are, first of all, how do disturbances impact our ability to assess reserve success? What roles can reserves play in mitigating the effects of climate change? And I'm going to talk briefly about two parts of this. The first one is what roles can they play by being virtue of a marine reserve in itself? And also, what roles could they potentially play if we combine them with other management actions? Sorry for people online, I just took a drink of water, if that sounded funny. So this is a huge area of research, and like I mentioned before, I just want to give you a bit of a snapshot of all the different work that we've been doing in this space. And I also want to point out that I mainly focus on fish species. I'm really interested in things like your blue rockfish, your kelp bass, sheephead. There's a coral trout, so that's from Australia. And the reason that I like to focus on fish species or harvested species is because they're one of the more influenced things by reserve protection. Like I've already said, you stop fishing, things come back. The other reason is because they're a really important ecosystem service. There are huge fisheries relying on these fish. They're people's livelihoods, and this is a really important thing that we can't lose sight of. So this is one reason why I really like to work on fish species, because it has impacts beyond just the importance of these things of what they are of themselves. I am a ecological modeler. So feel free to leave now if you want, but I promise there's no maths in this talk. But what we do is simulate the dynamics of fish populations. And we do that using age and size structure. Usually those models look like the way the models look is that they have populations of sedentary adults. So in this diagram, there's like a green box with one population and a red box with another. And the adults stay within those populations. So this obviously only applies to certain species and not big migratory species. And then those populations are connected through larval recruitment. And there's two types of larval recruitment that we think about. One of them is open and the other is closed. So if you have an open larval recruitment, it means that your babies come in from an external source and they're decoupled from the adult population. Whereas if it's closed, there's a coupling, there's a link going on. So the number of babies that are going to come in next year depends on the number of adults you had the year previously. So that's the difference between open and closed. And it just depends on what question we're asking as to which model we use. Because we're looking at reserve establishment, we need to put that in the model. So usually what that looks like is we run the model under a fish scenario, we add the reserve establishment, and then we look at the dynamics of the model, the population after the reserve is established. And then depending on which question we're asking, we just put in other events or factors such as disturbances. We might vary the fishing pressure, we might vary different aspects of the life history traits of the species that we're looking at. So that's sort of a general overview of the types of models that I work with. Okay, so now we've gone through that. Part one is looking at assessing reserve success in disturbed environments. So this is really looking at that accurate assessment side of things. And this really relates to monitoring. So giving us an understanding of, how likely are we to actually see things within reserves, and that can help us inform our monitoring. Do we need to monitor longer? Do we need to monitor different? Things like that. So first of all, I wanted to talk about the basic methods of how we assess marine reserves in the real world. So not using models or simulations, and this will kind of feed into what I'm going to talk about in a minute. So it's important that we just have a bit of an understanding of this. So in the real world, we go out and we collect data, and we use that data to assess whether a reserve is having an effect. And in this context, I'm just talking about reserve effects being rebuilding populations. Are these populations rebuilding or not? So we collect data, and we collect the data and compare that to some sort of control. So that control might be before the reserve is established. It might be outside the reserve. In this diagram, the reserve is the impact. Or it might be a combination of the two, which is your before or after control, or your kind of gold standard Backy design. So we look at those comparisons, and then we statistically test these comparisons to find out whether the reserve has had an impact or not. And most commonly for reserves, we see this sort of inside-outside design. And the reason for that is generally logistics. In the past, it's been really hard to know when a reserve is going to go in. We might not have been interested in really monitoring them at that point. So a lot of the data comes after the reserve has been established. But there are before, after, and Backy comparisons as well. So given that we've got these different types of comparisons being used, we really wanted to know how these different comparisons are influenced by a disturbance, when a disturbance comes in after a reserve is implemented. So it's not about picking which one of these is best to use. It's more about understanding how they're biased when a disturbance comes in after a reserve has been established. So to do that, to understand the impact of disturbances on these different approaches, we need to simulate the process. So if we look at a simple reserve establishment scenario, no disturbances at this point, what we do is we start off by simulating some sort of expected results. Sorry, there we go. And in general, if we're meeting that goal of rebuilding populations, we expect to see the simulated biomass increase over time. So that's the thick line is the average biomass, and then the individual lines are different variability. So because nature is inherently variable, we need to add some sort of stochasticity to the model. In this case, we're adding it through fluctuations in larval recruitments, the babies coming into the population. So what you're looking at here is that each individual line is an individual run or an individual simulation, and all of these lines together are a snapshot of the possibilities that could be happening in reality. And it's a spread of everything that's possible. So what we can actually do then is we can take a slice in time, and we can look at the distribution of those biomass, of those possibilities at that point in time. And then this allows us to make a statistical decision about how likely is it that we're going to see greater biomass in the reserve compared to the reference area. And this reflects a management decision of deciding whether reserves have increased relative to some sort of reference point based on the range of observable biomasses or biomasses that we've observed. Then what we do is we actually can quantify the statistical decision. And the way we do that is we basically look at the overlap of the two distributions. There's like 2 or 3 steps before we get to this, but I didn't want to bog you down with all of that. It uses looking at the probability of true positive versus false positives, and it allows us to quantify whether it's really likely, what the chance is that we're going to see these distributions overlap and that we're going to be able to not tell a difference or to be able to see a difference. So, okay, let me explain this. You've got high detectability when the two distributions are far apart. So this means that there is a sure thing we're going to be able to see a difference between the reserve and the fished area. When these two distributions overlap a lot, we've got what's called low detectability, close to 0.5, and that's a coin flip. So there's a 50-50 chance that we're going to be able to tell a difference between, we're going to see a greater biomass in the reserve, or a 50-50 chance we're going to see a greater biomass in the reference area. So we're not able to tell a difference very well. So I'm going to be using the term detectability for the next couple of slides, and that's just what it means. So when we establish a reserve, we put a reserve in, and this is in a model, so everything is exactly the same between a fish population and a reserve population. The only difference is you're not fishing in the reserve. When we put a reserve in, we expect to see this detectability increase over time as those two populations diverge. Okay. And that's for all our different comparisons that I introduced earlier. We've put in a confidence limit of 0.08 here. I'm going to refer to this later on, but what that really means is that above this level, we're pretty confident we're going to see a difference between those two populations. And this reflects a typical stats cutoff that there's an 80% chance of not failing to reject the null hypothesis. So that's why we chose that 0.8 value. Okay. So then now we can ask the question of how do disturbances affect this detectability? So what we did is we used the blue rockfish population, an open population. We simulated a disturbance. We did a widespread and a localized, but I'm only going to talk about the widespread results for today. So that's just where both the fished and the reserve have been affected by the disturbance. We varied reserve age and length, the reserve age that the disturbance happened, and length and strength of the impact. And that's just one example of what one of the runs look like where we have an impact such as a heat wave come in. And a heat wave is important to blue rockfish, I should point out, because it affects the kelp, which affects them, essentially, I think. My ecology is not that strong yet on temperate species. All right, so this is what our results look like, and I'm just going to step you through them because there's a lot going on here. But essentially, we looked at detectability. So remember, 0.5 is bad, 1 is good. We can tell a difference. And we looked at how detectability changes over the length of the disturbance. So your 1 to 5 years, and then we looked at how detectability changes from as you increase the strength of that disturbance along the horizontal axis. We also looked at how detectability changes when you have a young reserve or when you have an old reserve. And I should have pointed out, this is immediately after a disturbance. So a disturbance has come in, it's left, and we're looking at detectability that year after. And then we looked at it for the comparison type. So your backy, your before, after, and your inside, outside. And what we can see here really clearly is that your detectability is more biased when you have a high impact long disturbance, which we expect. And it's also more biased when you're using the before after sampling design. So don't take that as a don't use before after in this scenario, just take it as when we have a disturbance, we need to be aware of the fact that before after is more biased by a disturbance than the other comparison types. So what we can see here is that immediately after a disturbance, detectability drops off dramatically, and it can easily drop below our 0.8 threshold that I mentioned previously. So that remember that 0.8 threshold is where we're really confident in detecting an effect. So the question that I thought from this that is really more interesting is how long does it take to actually come back? How long does it take for us to be confident again that we can then detect a reserve effect after a disturbance has knocked that detectability right down? And I thought I'd show you some example how detectability changes over time, just for one scenario of a 3-year moderate disturbance. And I wanted to point out a couple of things are going on here. So your dotted lines are no disturbance, you expect detectability just to increase over time. And the colors are when the disturbance happens after the reserve is established. And there's a couple of things going on here. So just look at the inside outside for now. One scenario is you have detectability build up and then a disturbance comes in and it knocks it down. But that happens before it's reached that 0.8 threshold. So it knocks it down and it just kind of extends the time out that it takes for that detectability to reach that cutoff limit. Another scenario we have is a detectability has built up and then it gets above that limit and then it gets knocked down, it goes below the limit and then it has to recover again. And then the last example is that it's built up, but it's built up enough that when you knock it down, it actually doesn't drop below that threshold. So what we did instead of looking at individual runs is that we quantified the time from the end of the disturbance until it hits that 0.8 threshold. And I'll go through the axis again before I show you the results because there's a lot going on. So we looked at how that time, that years to 0.8 detectability changes when you have the age of the reserve along the horizontal, when you have the strength of the strength of the disturbance along the vertical. So low is at the top and high is at the bottom. When you have a short or a long disturbance of the two columns, and when you have different comparison types along the rows. And then the numbers are the years it takes following the disturbance to get back to that 0.8 detectability. And what you can see here is there's a lot of zeros. So this is that scenario where the disturbance actually doesn't really affect the detectability. It doesn't affect our getting to our 0.8 threshold. But then there's also numbers all the way up to 10. So what we found was that it can actually take up to a decade, 8 to 10 years after a disturbance for us to get back to being comfortable or confident that we're actually going to see a difference between the reserve and the reference area. But it might take as little as just 4 years if we have like a short, low impact disturbance. Reserve age actually doesn't matter much in this scenario. And I think this is because we're dealing with an open population where the recruits are decoupled from the adults. I should have pointed out too, this disturbance affects just the adults. So when you have the adults knocked down, it doesn't affect what larvae are coming back, so they can recover quickly. And your before after is your most biased comparison type in this scenario. There is more to this story, but I just wanted to give you this little snapshot. And just for you to take home from this is that disturbances affect reserve assessments. We know this, not surprising. But what we didn't know previously was it can take up to a decade following a disturbance to get back to being confident that we can tell a difference and making a confident reserve assessment. And in the case of disturbances, before after is going to be your most biased by the disturbance. So we just need to keep in mind that we might have to wait a little bit longer after a disturbance if we're looking at before after data before we're confident that what we're seeing is before we're confident we're going to see a reserve effect. So this is looking at how disturbances affect reserve assessments. And that's working, like I said, under the assumption that we're assessing whether reserves are rebuilding population or fish biomass or not, which is a fundamental question in reserves. And this question really stems from the fact that historically reserves were put in place in response to exploitation of our oceans, with the caveat that that's a white colonial perspective. There are traditional reserves that have gone in for traditional management, but essentially it's all been about sustainably making sure there's things there in the future. But then climate change came along. So we're now asking the question, okay, as we move forward, as climate change really begins to affect these areas and affect our health of our planet, we're increasingly looking for ways to mitigate and adapt. And given that reserves we know are a useful conservation tool, we're asking the question of what sort of roles can they play in the face of climate change? And this is really around that goals and expectations side of things, because we don't have clear goals and expectations. Reserves might not meet what we expect them to meet, and then that can actually undermine the good that they can do. And this is the second half of my talk, and I'm just going to give you a little bit of snapshot of the work we've been doing in this area. This is a really hot topic at the moment, and there's a really great new paper out that outlines, currently what we're thinking and what we know in this space and what reserves can do in the way of mitigation and adaptation. So they looked at the pathway through which reserves can contribute to climate change mitigation adaptation, And then they looked at the evidence that there's a reserve effect that's likely to support those pathways. And I will also point out there was a paper that Will was on that looks at this for California as well. So if you're interested in this space, just get in touch and we can give you some references there. But the point is that while we're pulling all of this sort of knowledge together and we're understanding sort of ways that reserves could be doing things, we're still really grappling with what this actually looks like in the trenches, what it looks like in the reserves, what does mitigation, adaptation, and resilience really, really mean? And for me, this is a question of what does this mean for fish species in the face of disturbances? And I put in a little bracket of those who rely on them, because I do look at yields and fishable biomass, but this is through an ecological lens, not a social lens. And obviously that's something we need to keep in mind moving forward as well. So what does this mean for fish? So as I've already mentioned and probably hammered a little bit too much already, reserves rebuild populations, we know that, but what they also do is that they rebuild the age structure of fish populations. So when you have a fish population, the age structure is truncated to the smaller ages or the smaller sizes. When you protect that population, it rebuilds out and you get more large, old individuals. And what this means is that you actually have a greater reproductive capacity. And theoretically, in the face of disturbances, this should mean that there's a greater capacity to recover from disturbance. So to recover quicker from a disturbance, a greater capacity to persist against really, really bad years. So there's a couple of different ways we could look at resilience or adaptation or mitigation, just depending on what you're interested in or what is more important, or look at it all together, I guess. So theoretically, we understand what might be going on, and we have some sort of theoretical evidence that supports this as well. So during my PhD that Will mentioned, I was working on coral trout, and we were asking the question of how fishery yields and fish biomass are affected by the rezoning. So in 2004, they increased the area in the Great Barrier Reef that was under reserves to 33%. And we just looked at what that means for coral trout, which are delicious. And they're really important because they're a live export out to China, which is one of our biggest trade partners. Anyway, that's a side note. What we did in this particular study is we looked at how variability, specifically long-term variability, so population bouncing around in time, how that long-term variability changes when you have a reserve. So think of this situation as you have lots and lots of multiple disturbances happening in time. Not necessarily big disturbances, they're just happening a lot. And what we found was that on the whole, when you have more reserve coverage, so going from the black to the orange line, you have a more stable biomass and you have a more stable yields as well. But the caveat to all of this is that the reserve placement, where you set the reserves, really, really matters. So we looked at biomass and yield variability under a couple of different scenarios. So we looked at the scenario where you put your reserves in areas that are more frequently disturbed, so areas that get lots and lots of disturbances. That's the orange dots. And we found that biomass and yield trade off, which is pretty normal for reserve scenarios. So if you put your reserves in disturbed areas, your biomass variability goes up and your yield variability goes down. So yields become more stable. It's good for yields. If you flip that around and you put reserves where there's no disturbances happening, then you get biomass variability, or biomass, sorry, becomes more stable, yields become less stable. So if you don't want to have to trade off as dramatically, what you can do is put reserves in a mix of both disturbed and non-disturbed areas, which are the green dots. But the trick here is that the reserve needs to be central and it needs to be close to the other reefs or the other populations. So in the Great Barrier Reef, we have patchwork of reefs and patchwork of reserves, which is different to what you get along the coast here, where you have reserves in a line along the coast mostly. So this is probably closer to like a Channel Island situation down in California, if anyone's familiar with that. So we're looking at the long-term variability where we have lots of disturbances happening. Another thing we can look at is what happens when you have a single acute disturbance. What do the dynamics look like then when you have a reserve versus not having a reserve? And One thing we can look at when we're looking at that sort of zoomed in perspective is we can look at the impact. So how much does a disturbance knock down a population? And we can look at the recovery time. So this was a recent paper by Will and colleagues, and they weren't looking at reserves specifically, but what they did look at is the post-disturbance dynamics of rockfish under different fishing scenarios. And what we can extrapolate from this is that if you have a low fishing pressure, it's kind of like having a reserve, versus if you have high fishing pressure, which would be your fished area. And the kind of take home from this was that when you have reserves, you have lower impact. So it moves to the left along the horizontal. But the return time, how long it takes to recover after a disturbance depends on whether you have an open or closed fish population. So remember, open, the babies are decoupled from the adults. The closed, the babies depend on the adults. So if you have a closed population, it's probably likely that your return time is going to decrease when you have a reserve. But if you have an open population, your return time is probably going to increase when you have a reserve. That's what I just said. So building on from this concept, Will and I are just sort of looking at this specifically in a reserve scenario now. And these are some really, really preliminary results for kelp bass, looking at what happens, in the first 10, 20, 30 years after a reserve is established when you have a disturbance come in. So we looked at the impact and the recovery time versus reserve age along the horizontal. And then we looked at different disturbance magnitudes, high, medium, low, and length of disturbance, 1 to 5 years. And, oh, sorry, I'm looking at the wrong little screen. No, that's right. Then we looked at reserves and non-reserves. So reserves are the solid line versus a non-reserve scenario. So for impact, we found that, reflecting the results of the previous study, when you have an impact in a reserve, the impacts are generally less. So in this case, higher numbers means less impact, which is a good thing compared to a fish scenario. But when you have, but the recovery time's a little bit more complicated. So when you have a young reserve, your recovery time is really, really quick. But as the reserves get older, your recovery time actually starts to increase. So it takes longer following a disturbance to recover if you have an old reserve. And I think the reason for that is that in this model, we're using density dependence. And when you have, when you're starting to get to your carrying capacity with density dependence, which is what you're doing in an old reserve, your population growth is lower. So your recovery time will generally be slower at that point. And then also we found that when disturbances were large or long, your recovery time is actually slower with a reserve than a fished area. So like I said, these are really preliminary results. And the general take home is that reserves might promote resilience. We think they're generally going to decrease long-term variability, decrease the impacts from disturbances, but the recovery time is dependent on the life history traits of the species, as well as the disturbance characteristics as well. So we really need to dig deeper into this and look at more what this also means for yields and fisheries as well. Okay, so the last little thing I just want to quickly talk about is that reserves do not exist in isolation. I really like this cartoon. It's a bit old, but it's from the North Atlantic, and it really shows that there's a lot going on in our oceans. We have reserves. We also have ocean development. We have multiple people wanting to use the ocean for multiple reasons. And there's a lot of ways that we're managing our oceans, and reserves is just one of those ways. And likewise, there are a lot of actions that we could take to help mitigate the effects of climate change in our oceans. And marine reserves or marine protected areas are just one of those actions that we could take. Another action we could take, which is a big thing along the coast here, is kelp restoration or habitat restoration of kelp. So this has been increasing over the last couple of decades, and we're seeing more success with this. And essentially the general premise of kelp restoration is you have a degraded area, you restore it, and then the ecosystem that relies on kelp then comes back, And this means that we have more fishable biomass, people are happy, everything's, you know, golden if it's successful. And it's a lot more complicated than that, but let's work under that premise here. So we wanted to know, can reserves enhance the benefits of this process of kelp restoration? And I'm just going to give you one of our results. There's more to this story, but one of the results is we looked at kelp bass. So kelp bass rely on kelp for recruitment. So no kelp, less recruitment, more kelp, more recruitment. And we looked at the population benefits and the yield benefits as you increase the area that you're protecting. So point 2 is you increase the area that is restored. So point 2 is you've restored 20% of the area. So this is under a fish scenario, and the more area you restore, sorry, this is when you restore in a fished area. More area you restore, the more benefits for both the population and the yield. But if you restore in an MPA, you get much more benefits for the population, but this comes at a little bit of a cost for the yield. And something that I thought was really interesting out of this is that for the same benefits, you can get the same benefits, but by using half of the area if you do that restoration within a reserve. So if you restore 20% in a fished area, you're going to get the same benefits if you only restore 10% in the reserve, which is important because kelp restoration is really expensive. So the more area or the less area you use, potentially the more bang for your buck you get. But like I said, this comes at a small cost to fisheries. And when I say cost, it's still better than not restoring. But if you restored outside, it would have been slightly better than if you'd done it in the reserve. And like I said, this is just part of the story, but the take home here is that pairing reserves with kelp restoration may be beneficial. Just depends on your goals. And this links back to what I was talking about throughout this talk, is that we need to set clear goals and expectations for what we can achieve with reserves. Okay, last little bit. I just want to take a step back, broaden this out, and sort of talk about where this work fits into the broader marine reserve world. So I started off the talk by talking about the 30 by 30 initiative, which is the goals to protect 30% of the ocean as well as 30% of the land by 2030. And this is happening all around the world. So Australia last week officially committed to the 30 by 30 initiative. And the government statement when they did this was that their plan was to halt and reverse biodiversity loss across our nation and put Australia's environment back on the path to recovery, which is a very ambitious goal, I think. And we're asking a lot of protected areas here. And we know protected areas can be really good conservation tools, but the premise behind the 30 by 30 is that if we have more of these good conservation tools, then surely that must be good, right? But I hope that you've kind of got a little bit from what I've talked about, or at least I've alluded to, that it's more complicated than this. There's a lot more going on. There's a lot more we have to think about. And I think about it like we're in a ship and we're trying to get to Treasure Island. Okay, and there's really complex waters to navigate between us and Treasure Island. And there's a lot of obstacles and dangers and hurdles on the way. And if we navigate really, really well, then this could present a really unique opportunity to reach Treasure Island, to reach the bounty. And it provides a unique opportunity to, support the conservation of our natural resources, but also provide benefits to those communities who are deeply linked to these systems, culturally, spiritually, or for their livelihoods. But if we navigate it poorly, then we could have really severe ramifications, especially for those people who rely so deeply on these systems, on these areas. Also, if we do a bad job of managing or protecting our oceans, if we don't set clear goals and expectations and adaptively manage with all the tools that we have, then reserves could potentially fail to deliver the benefits that we set for them and we expect from them. And this will severely undermine the good that they can do. So the work that I presented a bit today that we've been doing, and the work that my colleagues do, I see them like tools in this whole thing. So if we're going with our ship analogy, I think of our work like a compass and a spyglass. Without these tools, a ship can still sail through the water. It can still try to get to Treasure Island, but we can't, without a compass, without a spyglass, we can't see where we're going. We can't see what direction we need to head in, and we can't see what Treasure Island looks like when we get close to it. So these are the type of tools that help those people who are navigating, those people who are making the decision to make a really informed decision, and to help us make sure we get to the bounty and we don't end up on the Skull Island of Doom with all the volcanoes and the pirate. I'm pretty sure I'd rather be on Treasure Island, so let's make sure we get there. Thank you very much, everyone.
All right, everybody, thank you so much. We're going to open it up to questions, and we're going to start. Is there any questions online? All right, for folks online, if you have a question, feel free to put it into the chat. How about questions in the room? All right, here we go.
Really a great presentation, very entertaining too. A lot of the data that the graphs that you're showing are what? They're not the real numbers. They're straight lines and things like that. They're ideas.
Yeah.
And so, and the reserves, and I work on something that nobody likes that I'm trying to save. So it's like with fish or I don't know, kelp, the effort is not to kill them. And so a reserve, you can keep people off, or you can manipulate the space somehow. But conservation with climate change, like you're talking about, is something very different than space. And so in the reserves, it's not just to keep a place, I don't know, to restore a place. How do you navigate how many species there are, what kind of species there are, if they all interact, those kinds of things, and the spatial distribution of reserves? I mean, if you're going to look at climate change and let's say things move north and south, some things do, not all of them. And so it seems like there's something like climate change is so much bigger than trying to protect a space. Is there any way you can come to grips with how big that gap is?
Yeah. What do you work on? First of all,
Something really glorious and charismatic. It's called the mud shrimp.
Oh, okay. So it's not something like mosquitoes or something that everybody really hates. It's, yeah. That is a really, really good point. Reserves are not going to solve everything. They're not the magic silver bullet that we're looking for. They're just one of the tools in the toolbox. And sort of the questions that I think we really need to ask, and we're already asking moving forward, is how can we set reserves in the future that are going to that will do a better job than if we don't. So for example, if certain species are moving north or south, do we need to set more reserves? Do we need to move reserves? Do we set reserves, like in the case of the coral trout and the Great Barrier Reef, do we put them in areas that are really hammered all the time? So for coral trout, the population I worked on was on a group of islands just off the coast, and they're most impacted by flooding events. So you get flooding events that have massive runoff, and then that runoff hits the island, and it mainly hits, so you've got Australia and the islands, so it mainly hits the onshore reefs. So maybe it's better to put reserves in in there, or maybe it's better to put them in the more pristine, less disturbed areas, which is what I talked about. So it's really about how can we give them the leg up, not how can we save everything, because that's not going to happen just with reserves. How can we give everything the best chance we can with the tools that we have, because we've got to try something with what we have. Otherwise, it's doomed to fail, really. Yeah. But that's a really good point. This is the small thing in a big problem.
Other questions in the room?
Hi, thanks for your presentation. That was really great. I just had a question about when you were referring to the kelp forest restoration, and you talked about, I think the population would increase in the reserve, but the yield would decrease. And I was wondering if that has any timeline, or if you would expect that to change over time. The yield decreasing in the reserve, I guess.
Yeah, the yield decrease when you restore within a reserve. Yeah. I'm just trying to think back to that model. So that was, yeah, we didn't look at short-term dynamics, but my gut feeling would be no, it wouldn't get better over time. I think given the work that we've already done on transients following a marine reserve, so what happens in the years after the reserve is put in, typically you can see yields decrease and then they bounce back up. So I think you would still see that happening. Yeah, good question though. I'll think more on that one.
Other questions in the room? When you were comparing the different experimental designs for detecting changes in reserves versus not in reserves, you were looking at before, after, impact control, and then Backy. And it looked like a lot of the before after were very heavily biased to the point where like the Backy design didn't seem all that much stronger compared to just the inside outside. Is it something to do with the fact that the literature that you were pulling this from are like biased in some way? Is that they don't start early enough? Like, does there seem to actually be any sort of benefit to doing before after? Or do you think it makes more sense to just do inside outside?
Are you talking about in general or in the context of disturbances?
In the context of detecting changes due to disturbances.
Yeah.
Or detecting biological impacts of reserves in general, I think was when that came up.
Yeah. Okay. So that came up in general. In general, no, I don't think there's go with the Backy. If you have the money, go with the Backy, because that accounts for your spatial and your temporal differences. That's what Backy design is. In the context of disturbances, it's more about understanding how to interpret the data when you have the data. So if you just have data from before or after, you need to be aware that it might be more impacted by a disturbance than a Backy or inside outside. The reason I don't think a Backy was as impacted, and I'm going to look more into this actually, but I think in the model, but I think the reason the Backy wasn't as impacted as the before after, even though it has a before component, is because it's mitigated by the inside outside, that's why you have a Backy, because you're trying to mitigate for both inside, outside, and spatial. Yeah.
Okay, so I forgot to mention we have a bunch of students that are taking the seminar for credit as well. Can you just do a quick definition of Backy for us?
Backy is before, after control impact, and there's multiple different types of Backy as well. But essentially, it's your sampling before the impact, after the impact, and you're making sure you do that in multiple spaces in time. Multiple spaces, multiple areas in space. So you're doing it in the impacted area and outside the impacted area. So in a reserve, you're sampling the reserve before it happens, and then you're sampling it after it happens, and then you're sampling the fished area before the reserve goes in over there, and then continuing to sample after it. And ideally, you actually want to sample multiple times in space and multiple times in time. Sorry, the jet lag is really starting to kick in.
Perfect. That's just what I needed. We have a question online. Go ahead, Roseanne.
Yes. Does this narrative change for cold water versus warm water species? Have you looked into this narrative for diversity metrics?
Personally, I have not. I honestly don't know. I don't know the literature if they have or not. Sorry.
All right. Also, your contact information is on the
Yes.
screen. So if folks have other
Yep.
more detailed questions or want to continue the conversation, you can do that.
You can also contact me at my Oregon State email as well. I just put that one up there as that's just a more generic one.
All right. Last call for questions in the room.
All right.
That was a wonderful presentation. Thank you. So going back to the disturbance part, you had a long-term and a short-term disturbance. Are you trying other disturbances as well? And would you expect similar results?
Other disturbances in terms of?
Like you had marine heat waves, for example, but you also had cyclones and then like more persistent or short-term or persistent events.
Yeah. No. At the moment, no. I mean, yeah, this could apply to cyclones theoretically, like it could apply to any sort of pulse disturbance. The sort of consistent things is more what that long-term variability stuff was about. But yeah, we've tried to cover an acute disturbance that could last 1 to 5 years, 1 to 10 years. We are looking at that, or be of X, Y, or Z strength. And that can be applied to a lot of different scenarios. But no, back-to-back disturbances, we haven't looked at the close dynamics of that yet.
All right, last call for questions online. Go ahead, Roseanne.
Yes, this is a more general conservation question. How do we balance collecting more tools, research, and making concrete conservation decisions like protecting a particular area? We're talking about climate change.
Yeah, okay. Oh, that's a good question. It really comes, I think my perspective is it comes down to really clear goals. What do we want to achieve? And do we have the tools at the moment to achieve that? So it kind of comes back to the question I had at the start of, you know, marine reserves aren't necessarily a tool that we set out to go, oh, this is going to help with climate change, but it's a tool we already have, and it's a tool we already know a lot about. And it's a tool that we know a lot about theoretically, and we've done a lot of management on it. So it's about going, okay, well, can this tool then help in this scenario as well? But to do that, we really need to set really clear goals and expectations and be specific about what those mean, and keep checking up over time to make sure that they're meeting those goals. And then if they're not, what can we do with that? Yeah, because if we do keep trying to think of new things, then yes, that takes a lot of time and money and effort, and that, you know, swings that way of the balancing act.
All right, I'm going to hand this off to Will. He's started us off. He's going to wrap us up.
Thanks, everyone. Thanks, Jess. And I just wanted to say to everyone in the audience here that Jess, like I said, will be here for the next 2 weeks. She's in an office up on the second floor, so feel free to go by and ask her more questions about models and modeling and things like that. Also, if you enjoyed the amazing graphics in this talk, I want to say that Jess is too modest to say that she also has a graphic design and scientific illustration business that she operates, and does amazing infographics and sort of other illustrations. And I think you can find a link to that in her Twitter bio if you check there. So thanks very much, Jess. That was really lovely.
All right, thank you so much, Jess, for being here, and for everybody. Hope to see you next time. Thank you. Okay, for folks online, I'm going to end the presentation. Thanks for being here.